Hi, welcome to Exam 3 written portion. This is by Katie Jones, and so let's begin. Alright, number 1. So this question concerns the human disease that we have discussed in this section of the course. Below is the group of terms that we have learned about recently, and it's triploid, inversion, deletion, monosomy, duplication, aneuploidy, translocation, trisomy, and non-disjunction. So 1A. Which of the above terms is associated with Patu syndrome? And I say it's aneuploidy, trisomy, and non-disjunction. B. Which of the above terms is associated with the conventional form of Down syndrome? And it's aneuploidy, non-disjunction, and trisomy. C. Which of the above terms is associated with Turner syndrome? And it's aneuploidy, monosomy, and non-disjunction. And 1D. Which of the above terms is associated with Cru-du-Chat syndrome? And that's deletion and translocation. And 1E. Which of the above terms is associated with type A hemophilia? And that's inversion and duplication. So question 2. Examine the E. coli DNA sequence shown below. So we see 2 primers and the sequence. And the bottom strand is the DNA strand. So yeah, those are the 2 sequences. And so that will be used to synthesize the lagging DNA strand. Note that this newly synthesized DNA strand will be synthesized as Okazaki fragments. So this strand, the 3' to 5' strand, is a lagging strand. So when you indicate the polarity of the primers on the image above, and its primer A is 3' to 5', and primer B is 3' to 5'. 2B. What is the DNA sequence that will be synthesized between the primers? And we need to indicate the polarity. And so what we have here is the indicated the new synthesized strand, and it's 5' and 3'. C. Which direction, right to left or left to right, is the replication fork moving as the starter strand is being synthesized, and is left to right? And D. Which of these 2 primers will synthesize first, and it's primer B? So suppose that the individual is heterozygous for the following change in chromosome structure, and we see an inversion here, and we see the chromosomes. And so what is the allele and centromere composition of the 4 products of meiosis if there is a crossover between T and U? And so here we see a percentric inversion, and so shown here is the chromosomes. So question 4. Suppose that the DNA molecule shown below is a portion of an E. coli gene. This E. coli gene is transcribed from left to right, and the +1 side is shown below, and we see the 2 strands here. So A, which strand, upper lower, is the template DNA strand, and which upper lower is the coding DNA strand? So top is coding and bottom is template. B, what is the sequence of the mRNA molecule produced from this DNA molecule, and that's shown here. And 4C, in which direction, left or right, along the coding strand relative is the +1 site? Would the -10 and 35 sequences be located? And it's right of the +1 site. D, in which direction, left or right, along the coding strand relative to the +1 site would the terminator be located? And it's left of the +1 site. 2. And that is all, and thank you for joining us.